Nerve tissue. Remember, this is one of the four basic tissue types, and no tissue is like this, right? You have connective tissue, you have epithelial tissue, you have muscle tissue. But muscles really can't work, especially skeletal muscles can't work without nerve innervation. Yeah, I mean, except for the heart. The heart has its own built in conduction system, but nerve tissue is very specialized, very electric. When you hear nerve tissue, you got to think electric. Electric. What does that mean? That's all the sodium, potassium, chloride, all those ions are running in and out of the membrane, changing the electrochemical gradient, causing action potentials. Well, first depolarization, action potential, and then repolarization on neurons. So you should be familiar with all that. And I'll show you how the neuron is designed to do that. So, of course, the functional cell, the parenchyma of the nerve tissue is the neuron. So we'll look at the neuron, and then you have glial cells. Glial cells are supportive cells, or neuroglia, sometimes they're called. So we're not going to really look at too many glial cells, but I think you have to remember, remember CNS, glial cells, you have your oligodendrocyte. And that is the CNS glial cell that produces myelin. So we're going to have to look at myelin. And that's that lipoprotein that surrounds the axon that speeds up conduction through saltatory conduction. So conduction has something to do with that electric potential and the action potential. So impulse, conduction all kind of mean the same thing. So myelin speeds that up. In the PNS, the peripheral nervous system, the myelin producing cell is Schwann. Schwann. Right? So Schwann cell is only in the PNS, around the axons, forming myelin. And they look a little bit different. So let's look at this thing. You've seen this before. You had to, because you're talking about nervous system. You're done with that. Moving on. All right. So I guess the place to start is basically this whole big structure right here. This whole thing. Yeah, makes sense. So that whole part is called the cell body, right? Cell body, neuron, cell body. Also known as the soma. The soma means body, from what I've heard. So you can call it either one. Just don't call it centrum. Call it soma or body. So information comes into the cell body, and usually by these tree-like or branch-like or tree-like appendages called dendrites. Dendrites are trees, or branches, I should say. Dendrites. So usually the information is coming in from another neuron to the dendrites. So this is the direction coming into this neuron. And you could also have information coming to the soma too. And you could have information coming to the axon too. But typically synapses, and you know what synapses are, that's the gap connection between two neurons or between a neuron and a muscle or a gland. So most synapses are axodendritic. You know what that means, right? Dendrites. So dendrites are usually incoming information. Where does the information come to? The cell body. The cell body is like any other cell. It has organelles like a nucleus. It has a nucleus. It's not like a red blood cell. It's anuclear. And the nucleus has a lot of things going on in there, like DNA, right? You have your DNA. You've heard of that. Deoxyribose nucleic acid. And that's our template for our genetics. So our genes are full of, our neurons are full of chromosomes, right, in the nucleus that have genes on them that build proteins. You gotta build the structure of a neuron, right? So proteins are important. So within that nucleus, this little ball, don't get these two wrong, everybody gets this wrong, because it's just a couple of letters that are different. This is the nucleolus. See a couple of different words, letters there? Nucleolus is within the nucleus, because the nucleus has its own membrane. It's porous, it has nuclear pores. It's called the nuclear envelope. So the nucleolus is basically building ribosomes. And you know that in the cytoplasm, the ribosomes come out of the nucleus, and that's the site of protein synthesis. That's how we build proteins. That's what we're all about. Our structure is proteins. Remember, amino acids are the building blocks for life, but for protein specifically. Yeah. So the cell body and the axon, technically, the axon has this too. So, you know, the cytoplasm is anything between the nuclear membrane and the cell membrane. So remember this, and this is why it's so excitable. Every part of this neuron has a biphospholipid membrane. That's excitable everywhere, all the way down to the end. Over here, here's the end. So the cytoplasm has things like ribosomes and proteins, but in the neuron, we call the ribosomes, we call them Nissl bodies or Nissl substances. I just like saying Nissl because there's only one vowel in there. Isn't it cool to say Nissl? It's like Schwann, Nissl. I like saying Nissl. So Nissl is the chromatin, basically, the proteins and the ribosomes where proteins are being built, because you need structure here. You need microtubular proteins that give the neuron its structure. Come on, right? And this PowerPoint doesn't point it out, but there's a ton of mitochondria. And mitochondria is everywhere in the neuron. All the way down here, too, you have mitochondria. So there's cytoplasm throughout the whole neuron, not just in the cell body, because the whole neuron is surrounded by a membrane. I told you that. I don't mess around, right? So this part, D, is basically called the axon hillock. Don't worry, I'll tell you what the axon is, but you know that already. You've taken this class already. Axon hillock. Hillock is, hilum is anything that's coming in or out of a cell or an organ. In this case, it's just coming out of the cell body. So you have an axon hillock, then you have an initial segment of the axon. And in here is where all those graded potentials are going to be summated. So I like to call this the trigger zone, like between the axon hillock and the initial segment of the axon. So all this yellow stuff all the way down, and here too, this is a collateral axon. This is all called axon. So technically, I think E is just saying axon. Sometimes you hear nerve fiber, neuron fiber. That's because axon is like a stringy fiber that goes away from the cell body. Remember, the direction is going this way. And that's called anterograde conduction. Because look, all these neurons coming in, depolarizing chemically and electrically, depolarizing the dendrite membranes. And then all the information is brought into the cell body, which is summated in the axon hillock area, an area called trigger zone. Trigger zone is not an anatomical place, it's physiological. So that conduction will continue. And remember, these things, let's see, where do we point these out? Like anyway, this cell, and you see a bunch of them, this is our friend, you know, the dead neurologist named Schwann. Schwann cell. I think your book calls them neurolemocyte. Let's write that out. Neurolemocyte. I don't care. So if your lecture instructor might have that in the book, neurolemocyte is the same as a Schwann cell. But I like saying Schwann, so I always say Schwann. So you remember last slide I told you that this PNS cell, Schwann, makes myelin. So this whole thing right here is the myelin sheath. Myelin sheath. What the hell is a sheath? Well, imagine like you took like a bathroom towel, right? And you wanted to keep your arm warm and you wrapped it around and rolled it up like a yodel. So you keep wrapping it till you get that sheath, which sometimes is called lamellated sheath. And don't forget, this is not a Schwann cell, is a glial cell. It's not a neuron. So it has a nucleus. So this thing right here, this little football in there, is the Schwann cell Count how many times I say Schwann, Regime. Schwann cell nucleus. Yeah, right. Then, do you notice that there's a gap between the Schwann cells of the myelin? That's not an accident, right? Because we want this conduction to jump over this neurolemma from node to node, and then get back to the terminal. So that's called saltatory conduction. It's much faster than if there wasn't myelin. So every time that conduction reaches that gap, you're going to have depolarization, action potential, repolarization. That continues down to the end of the axon. So I can't fit it in here, but that gap, right, the gap between the myelin, between the Schwann cells, and you got to say this with a French accent, node of Ranvier. Okay, so that's the gaps between the Schwann cells. So nodes of Ranvier. They might call them neurolemmacyte nodes or gaps in your book, so it might be a little different name. But I like to say Ranvier, again, node of Ranvier. So you'll get it right either way. When I put it on the practical and I'm pointing to the gap, you say node of Ranvier or whatever your book says. All right, but when you write it, please say it with a French accent, then you'll get extra credit. So over here, now there's still the axon. So now we have these, they call this telodendria, which is like the arms of the axon, the end of the axon, really, telodendria. So I don't really like to say this because it kind of confuses you with dendrite, because it's all, dendra means like branching. So the axon branches too into these little telodendria, right? But this is the magic area right here. And this is a bunch of different names, this end of the axon. So let's make it simple. I mean, I think just make it simple. It's called the axon terminal. That makes sense, right? Grand Central Station, Grand Central Terminal. So the axon terminal is what that's called. But you can call it the synaptic end bulb. You can call it the Bouton axon end bulb. But you hear all these terms, bulb, terminal, right? Because this is the place where the vesicular neurotransmitters are stored. You know the story. If an action potential reaches this axon terminal, synaptic end bulb, then you're going to have calcium, voltage-gated calcium channels are going to open. Calcium is going to bind to those vesicles within the bulb, and it's going to release neurotransmitters like acetylcholine, norepinephrine, dopamine, serotonin, GABA, glutamate, some glycine, and all the other ones you've learned. So this is it. This is pretty much, I think, all the parts on your list. So myelin sheath, axon terminal, axon, dendrite cell body, neuron. You know the neuron. I'm just going to show you a couple of pictures on the slide. Again, I'm recording this, so what I say here is important because I'm kind of following your checklist. But there are going to be other slides to help you with your neuro. So this is a cross-section of the spinal cord. And notice this darken there in here. Looks like an H, right? We're going to do the spinal cord later, but this is gray matter. Now, you know what makes gray matter gray and what makes white matter white, because here's all white matter in the spinal cord. All right, so gray matter is cell bodies. Cell bodies and maybe unmyelinated axons. Makes sense. There's no myelin. There's myelin when you open up a brain or spinal cord, it is white. Those tracks are white. Those white areas are white. But gray matter is cell bodies, right? White matter is myelinated axons. That's simple. Who makes the myelin? You know, Schwann cells in the PNS, oligodendrocytes in the CNS. And you know the other glial cells, but I don't see them on here, my list. So I'd love to talk about astrocytes and microglial cells and ependymal cells. And I might mention that later. and also satellite cells, but we just really got to know that Schwann. That's pretty much it. And you have dorsal or posterior, anterior or ventral. Remember that anterior is also known as ventral, just to keep that in mind. Posterior is also known as dorsal. Because as far as I know, we're bipeds. We walk upright, most of us. And like a sheep is a quadruped. So you got to know his dorsal side is ventral side, or her dorsal side, ventral side. So it's kind of comparative anatomy. We say anterior and posterior, but mammals overall, including the porpoises and dolphins, you have to say dorsal and anterior. So get that right. I like this. I mean, I think this is a good thing to go through before your neuro exams, just to go through. Chromatophilic, I like to say Nissl. Neurofibril node, I like to say node of Ranvier. Right? Probably the next page has the other thing. Teledendria. You know, I like to say the axon terminals at the end, or synaptic end bulbs, and the teledendria are really the spiky things that are leading to the end bulbs, because that's the way it looks. And here's all your glial cells. Again, I'm probably not going to ask you this because you got your hands full with the cranial nerves. When I get to the cranial nerve tables, I'll go through that slowly and we'll kind of tell you what's important. Look, neurolemmacytes, but you know, right? Victoria, you know I want to say Schwann over and over. So satellite cells, of course, are PNS. These are the PNS ones, and these are all the CNS, glial cells. And I'll tell you right now, there's 20 times more glial cells in this nervous system than there is neurons. You might get that question. Who knows? Who knows? This is a histology. I don't know what's going on here. You're not going to know this. This is too much. Right? But you remember from earlier in the lab, you knew what a neuron looks like. So, because this is the neuron. This is the cell body of the neuron. And this is part of the axon here. Because these are astrocytes. I'm not going to ask you this, but it's kind of cool. And astrocytes, you know, are important for the blood-brain barrier. So the astrocytes look like stars. Remember, they're only in the CNS, and they connect through these foot parts, or these foot appendages that kind of latch on to the blood vessel. This is a capillary in the brain. Yeah, so the astrocyte, that's all that's really showing. I'm not going through action potential with you unless you want me to. I'll go through this, but again, I'm not going to ask you in lab about action potential. Brain and cranial nerves. Guess what, though? The brain is CNS. Remember, the brain and spinal cord are the central nervous system, but cranial nerves are PNS. They're outside the brain and spinal cord. But know this, the cranial nerves are basically, there's 12 cranial nerves. Let me just tell you this, there's 12 pairs of cranial nerves, right? And you got to write them like this, like CN, crane. You don't have to, but you could say cranial nerve 1, Roman numerals, right? Cranial nerve 7, looks like this, Roman numerals. So they're paired. But the nucleus, or the nuclei of the cranial nerves are in the brain. And 10 of the 12 cranial nerves have their nuclei in the brain stem, which is the midbrain, the pons, and the medulla oblongata. I like saying medulla oblongata a lot too, right? So the top two, like C1, CN1, and cranial nerve 2, do not have their nucleus in the brainstem, which you'll see. Very simple, those two. But the rest of them have their nuclei. What's a nuclei? Nuclei is a group of cell bodies in the central nervous system. A ganglia, or ganglion, singular, is a group of cell bodies in the PNS. You get it? So cranial nerves, although the nerves themselves are peripheral, the nuclei are in the brain. The brain. Okay, and we're going to have to go to APR with this because I'm not really fond of this PowerPoint overall. Like, I don't think it's that easy to look at all the pictures in one place, so we're going to have to spend a little time together in the APR. But let's get the names down, right? So this is if I took a knife and I cut this part of your head up, right? So you see the hair shaft. Remember that? And you have the stratum corneum, and the stratum granulosum, and the stratum spinosum, the stratum basal. So you have the epidermis, dermis, hypodermis, and then you get to the covering of the bone, which is periosteum. All right, then you have the bone. Remember, these are mostly flat bones with trabeculated, chewy center there, and the cortex that is more compact bone. So now, here's the coverings of the brain. And the coverings of the brain are called meninges. That's kind of a cool word to say, but meninges, right? Those are the coverings. Now, it's also the spinal cord too, but they're just showing you the brain. So I have to point one thing out that's different between the brain and the spinal cord as far as the meninges go. It's only one layer. So the top layer, outermost layer, is the dura mater. And that means tough mother. Like my mother's a tough mother, dura mater. So that's the outermost. But notice in the brain, you have two layers. You have the periosteal layer, which is closer to the bone. That makes sense, doesn't it? Periosteum, right under the periosteum, you have the periosteal layer of the dura. I'm friends with the dura, so I just call it dura. I don't always say dura mater. Right, so this is the periosteal layer. Meningeal layer is closer to the brain tissue. And that's the meningeal layer. And that'll continue all the way down around every part of the brain, really, and every part of the spinal cord, too. So the spinal cord only has one layer of dura, and the brain has two layers of dura. So let's just deal with the dura. Right? Now, there's like blood vessels above the meningeal dura, and this one is good to know. This is the superior sagittal sinus. This is blood. This is blood, not cerebrospinal fluid, which we'll talk about later. Right? Because you'll see this structure, and you learn this in lecture. The arachnoid villi is what brings cerebrospinal fluid back into the blood, because this is veins. So whenever you see sinus, it's usually a vein. Not always, but it's usually a vein. And veins bring deoxygenated blood back to the heart. So the CSF is going to be reabsorbed back into the blood via these structures called arachnoid villi. Just wanted to point that out, right? So, you know, in the spinal cord, you'll see there's a space, and this is more in the spinal cord, but there is this small space in the brain too. So above the dura mater, between the dura and the bone, is a space called the epidural space. Like some people may have heard of an epidural injection of anesthesia or anti-inflammatory into the epidural space. So that numbs the nerve roots, really. Epidural space is an area where you can inject anesthetic, especially in the lumbar area. Not so much in the brain, but I just want to point out this space as I'm doing the coverings. Right? Epidural, like when you're delivering a baby, right? You get an epidural injection. My mother didn't have that because I was 22 pounds when I was born. So my mother is a tough mother. She's a dura mater. She didn't need the epidural. Probably used whiskey though instead and cigarettes. Okay, so now the next layer below the dura is the arachnoid mater. Arachnoid. Now the arachnoid mater is kind of spider-like. That's why I call it arachno. It means like spidery. So there's a lot of blood vessels involved in the arachnoid mater. Yeah. So I'm going to tell you something important about the arachnoid mater in a second, but let's go to the space first. The space between the dura and the arachnoid is called the subdural space. Okay. Now, if you have a head injury, right? So you get whacked in the head, you know, you could have a subdural hematoma, which is blood pooling in the space between the dura mater and the arachnoid mater. And that's a heavy bleed. There's a lot of arteries above in that area. bring blood into the brain, really, and veins, but definitely a lot of arteries. So the next layer, and this one is like the tightest one, right on the brain tissue. Let me see, like you see this little purple line here. That's what it is. See that purple line that's covering the brain? That's pia matter. So that's really a tight covering. Like if you order a brain on Amazon, or even a cell phone, like compare, like you get a cell phone from Amazon, they have like this big bag outside. So the dura mater is like a bag, very dense, regular connective tissue. In fact, the arachnoid matter kind of sticks to that if you're cutting up a real brain. You know, then that's a dead brain, of course. And then the arachnoid matter is kind of part of the dura. But the pia matter is like a tight cellophane covering over the brain. Like you peel it away with a tweezer. So the pia matter is like tightly bound to the brain tissue itself, and the spinal cord too. Right? So as you would know, this would be called the subarachnoid space. Now, the subarachnoid space is kind of important for one thing. The subarachnoid space is where some of the CSF flows, cerebrospinal fluid. Some of it, because you're going to find out that cerebrospinal fluid is housed in these containers or spaces within the brain called the ventricles. So here's the pia mater. You can see that there. Okay, so that's pretty much it with the layers. And again, APR, it's okay. It's okay. I looked at it yesterday. It's okay. But I'll show you what some of the best ways to look at the meninges on the APR and which structures to highlight. Okay. So again, meninges, you've heard of meningitis, right? You get a bacterial meningitis, which is really bad condition of swelling and inflammation and infection and immune response within the ventricles, within the subarachnoid space, and this is a dangerous condition. Bacterial meningitis. Viral meningitis, not as bad, but still is painful, of course. And you might not even know you have viral meningitis because it kind of is self-limiting. But bacterial meningitis, you're in trouble. Okay, so if you have any questions, please ask. So let's just look at, again, since I cut part of your brain out, let's not waste this brain. Let's take a look. Now you see this, I don't know what color that is. I don't know, what do you say? Tannish, but it's really gray. This is gray matter. And this is the outer part of the brain. So that's the cortex, right? Cerebral cortex. Cortex means bark, right? Cortex means bark. Like this word here means bark. Not bark like a dog, but bark like a tree, right? The tree bark, the outer part. And that's all gray matter. So what do you think is in there? There's cell bodies and unmyelinated axons. No myelin. But here's where all the myelinated axons are. Incoming and outgoing efferent and afferent neuron axons that are myelinated, moving around the brain and moving in and out of the brain. Right? So here you go. Here's some words on this. I probably won't ask you a lot of the words. I might ask you about the subarachnoid space, though. So you know that's important. The septa, I probably won't ask you because I have to show you pictures of that. And the diaphragma sella, I have to show you pictures of that to really discuss that. But this is the blood flow and venous drainage. So there's arteries coming in, they're not showing you that. So again, you remember the superior sagittal sinus when I cut your head? That's the one that has the arachnoid villi in it. And there's other arachnoid villi, not just in the superior sagittal sinus. But this is draining all the deoxygenated blood from the brain, not just the CSF. You know, the arachnoid villi will reabsorb the CSF back into the blood. And that'll drain, you know, remember it's going this direction towards the heart, internal jugular down to the brachiocephalic vein and then into the right atrium of the heart, which is going to go to the lungs. So I'm not going to ask you a lot of questions about blood flow. You're going to do that in A&P2 extensively. So don't worry about this so much. Know about the spaces, though. What about the spaces? The epidural space, the subarachnoid space, and the subdural space. There's a the subdural, of course. Now, the subdural space, or right up below the dura, that's it's veins, right? It's mostly veins. So that the scary thing about the subdural space and head injuries is that you can get whacked in the head or have a head injury, and you can go to the hospital, have a CAT scan. You could see, like, say, a 1 centimeter hematoma in the subdural space on your MRI, 1 centimeter. But veins they work under low pressure. They don't, spill blood out so quick. So that bleed can continue for a week, 2 weeks, 3 weeks, and then the hematoma in 3 weeks after can become 3 centimeters or 4 centimeters, 5 centimeters. And that could really take up space in your head and affect your brain tissue. So that's kind of just why we're interested in these subdural spaces. Okay. Whatever. So again, you could practice with this. I'm going to go over to APR, talk about this, and you can use this if you like, just to go through the parts. Remember, they took the skin away, though. Notice that? You just got the both sides of the periosteum, and then the periosteal dura and the meningeal dura. These are arachnoid villi, and you can see the subarachnoid space. So this little line here is the arachnoid matter, and the pia mater is the tightly covered around that cerebral cortex and invaginating into the sulci, right? We're going to talk about this later. This is called, this little groove, the little grooves are called sulcus. Sulci is plural. And the hills on top of the brain, they're called gyrus, or gyri is plural. So just to point out some important things there. So know the layers. This is a pretty good picture. This is not a pretty good picture. So again, I'll go to APR. I'm not going to give you a mid-sagittal view like this to look at the meninges, but I will do that for the brain. You see this little squishy stuff down here? It's part of your brain, right? This is your cerebellum right here. Cerebellum. This is your pons, and this is your medulla oblongata. Yeah, but we'll see that later. So I'm not going to use this to name or test you on meninges. No way, no how. This is kind of cool, but maybe a little confusing. I'm going to have to go to APR again now because you don't know the brain parts. But the cavities that contain CSF, cerebrospinal fluid, just so we're clear on what CSF is. CSF is fluid. It's mostly water, of course, like any other fluid, but it has, lipids. It has some sodium. It has a little bit of glucose. We're not exchanging nutrients, though. So the fluid ultimately within the brain in these cavities basically are protecting the brain, cushioning the brain. Very little nutrition, but there is some. Okay, so it kind of keeps the brain tissue in place because the brain is very light, right? The brain is not a heavy. If you take a brain out, a live brain, it's not more than like 8 grams. So the lateral ventricles are on both sides on the top of the brain. And then you have the third ventricle. So there's 2 lateral ventricles, right and left. There's 1/3 ventricle. I'm just showing this for later. I would never test you on this picture. And there's 1/4 ventricle. So between the 2 lateral ventricles and the third ventricles is the interventricular. Inter means between. Foramen. It's really a canal, though, but they call it foramen. So the CSF is draining from the ventricles into the third ventricle, the lateral ventricle, to the third ventricles via this passageway called the interventricular foramen. And then it drains further down through a cerebral aqueduct. Now that's kind of important anatomically, which you'll see later. Cerebral aqueduct, also known as the aqueduct of Sylvius. That's a V. Sylvius. Okay, into the fourth ventricle, what you're going to see. So those are your ventricles. So there's 4 of them total. I don't care about the telencephalon. You probably might have learned that in lecture in your textbook. I don't care what the mesencephalon is for you, for lab, or myelencephalon. But you're going to need to know what the diencephalon is. So be familiar with that word for later. And of course, you got to know what the spinal cord is. And the central canal is a continuation from the 4th ventricle into the spinal cord. So this goes all the way down the central canal. It's a passageway of cerebrospinal fluid. But remember, I told you before that the cerebrospinal fluid also flows through the subarachnoid space in the brain, right? In the brain and in the spinal cord. So cerebrospinal fluid is found in 2 places. One is in all these ventricles, and the other place is the subarachnoid space in the CNS, brain and spinal cord. Guess what? Another table. I like these tables. Lateral ventricles, 3rd ventricle, the cerebral aqueduct, and the 4th ventricle. Remember, there's the interventricular foramen is between the lateral ventricles, and there's 2 of those. 2. Okay. So I can show you the ventricles right now. Now, this is a classic picture. This is a mid-sagittal head of the nerve tissue. And you can see pretty much everything except for the cerebral lobes here. So I just want to point out a few things on this picture, again, before we go to APR and go through your list. So this is a mid-sagittal brain. And I'll tell you this, the brain has 2 hemispheres, 2 sides, right and left. So most parts are paired, have a companion right or left side. So you can see the blue is going to be that sagittal sinus, or they're veins, basically. All of this in here, all this folds, looks like a spongy looking stuff like cauliflower. That's pretty much that's your cerebrum, right? That's your cerebrum, which is the largest part. Cerebrum. And we're going to go through the lobes of the cerebrum. There's 5 lobes to the cerebrum. And that's the largest part of your brain. 83% of your brain is cerebrum. That's where the cerebral cortex is, the gray matter. And then there's white matter as well. So I told you there's 2 hemispheres, right? And I don't see where the arrows pointed to this structure, but this structure right here, this is not connective tissue. This is actually nerve tracts, myelinated nerve tracts, all of this big thing here. And that's the connection between the left and right hemisphere of your brain. Cerebral, mostly. This is called the corpus callosum. I think that's a good place to start. Big body. So corpus callosum. And there's a smaller connection down on the bottom, which also has tracts. Now, the crossover from left to right, the tracks that cross over from left to right in any CNS structure is called a commissure. And you'll see what that means later. Can't really see that in this picture because this is the right side of the brain in a mid-sagittal cut. So, but this part is called the fornix. The fornix, like a little alcove there. And you'll see fornix in reproductive as well, because it really just means an area or an out-pocketing within a structure. Now this area right here, between the fornix and the corpus callosum is actually a membrane that separates the 2 ventricles. And that's called the septum pellucidum. So the septum is anything that's a septum is separating 2 compartments. So in this case, the septum pellucidum is separating the right and left ventricle where the CSF is. So if I'm pointing to that and you said ventricle, you'd be right. It's hard to say what I would have to say. Name the membrane, and then you'd say septum pellucidum. If I said name the cavity, you have to say lateral ventricle. Right? In this case, it's the right lateral ventricle, but that's not that important. So I think that's the first thing that jumps out at you when you see this is this area, the cerebrum, of course, and you see the hills or gyri, or gyrus. I'll put gyri because that's plural. And the grooves in between the gyrus are called sulcus, or sulci. Sulci is plural. Okay, and some of those are important. All right. So just to get you familiar with this area right here, there's 3 parts to this. All of this right here in the middle, deep brain. Now we're getting deeper into the brain. The cerebrum, of course, is relatively new over the last 140,000 years. I've only been here for 139,000 years, so I wasn't there when it started. But this is old brain. So you can't say old brain, but it's got 3 parts, and that's called the diencephalon. Hold on. Diencephalon. Okay? And it's got 3 parts, which I'll show you later. Okay? Then we have a lot of parts here. There's like 3 parts right here, all the way down to here, say. And all of this, it's 3 parts. Midbrain is up here, pons is here, and the medulla oblongata is here. That's called the brain stem. Okay, and we'll go through the 3 parts of the brain stem. This thing right here is not part of the cerebrum. Don't make that mistake. That's over here too, sorry. They cut it in half. So this is the little cerebrum, but you can't call it little cerebrum. You call it cerebellum, which means little cerebrum, right? Cerebellum. So don't get the cerebrum mixed up with the cerebellum, because you don't think conscious thought with your cerebellum. You do that up in the cerebral cortex. Cerebellar cortex is more about balance and muscle contraction and equilibrium, motor. A lot of motor things there in the cerebellum. And notice the white matter. You see the white matter has like branches, and always, and every practical, we always put the white matter of the cerebellum because it looks like a tree of life. Don't say tree of life though, although I have to give you some credit. But you won't remember, I mean, you won't forget the name. The name is called arbor vitae, a tree of life, of the cerebellum. So you hear arbor vitae, you think cerebellum. All right. And if I ask you what layer this is, this white layer, that's going to be the pia mater. Yeah. All right. And here's your sphenoid sinus of your skull. Right, here's a frontal sinus of your skull. And remember the sella turcica of the sphenoid bone? It's right there. So that's housing, not housing, it's kind of forming a saddle called the hypophyseal fossa for the pituitary gland. Pituitary gland is ventral, right, on the bottom of the brain. Now this is an upright brain, right? You see quadruped, it's a very vertical. Well, you see the sheep. The spinal cord goes this way. So the pituitary gland is on the bottom of the brain in the adult, I mean, not in the adult, in the human and in the sheep. So you'll learn all about the pituitary gland. But remember, the pituitary gland is not a nervous tissue, technically. The pituitary gland is endocrine, and you're going to find out that's a fun bag of hormones between growth hormone, thyroid stimulating hormone, prolactin, adrenocorticotropic hormone. It's just a gonado, LH and FSH, a luteinizing hormone and follicle stimulating hormone. It's just a fun place. And then that's just the anterior pituitary gland. The posterior pituitary gland has more neurological connections to the hypothalamus, which is here. You're going to find out. And the posterior pituitary gland doesn't produce anything. Sorry. It just stores and releases oxytocin and ADH, which is vasopressin or antidiuretic hormone, right? So the connection, I think this is something we may ask though, the connection between the hypothalamus and the pituitary gland is a stalk. I mean, you can call it pituitary stalk, but why don't we use another cool name called the infundibulum, which means stalk. Infundibulum. So that connects the pituitary gland to the hypothalamus. Fun, it's fun. It's fundibulum. It's a fun bag of hormones that is controlled by the hypothalamus, which is part of the brain diencephalon. Please don't get that wrong. That's really important. Because they used to call the pituitary the master gland, but the master gland has a master, and the master is the hypothalamus. Remember all those feedback mechanisms you did? Well, the hypothalamus controls a lot of that, along with this guy down here, the medulla oblongata. So the hypothalamus and the medulla oblongata are very old parts of the brain that have very visceral and autonomic functions like heart rate, hormone release, and respiratory rate, and hiccuping and coughing and throwing up, and all these things that I did 139,000 years ago when I wasn't thinking. But now we think more, so we have a cerebrum and a prefrontal cortex, which you'll find out later. Now, posterior to the, or dorsal to the diencephalon, you have another gland called the pineal gland. So get these two right, because pineal gland is in the back, or the dorsal side, or the superior side behind the diencephalon here. And the pineal gland secretes a hormone. So the pineal gland is endocrine, not nerve tissue. So the pineal gland secretes melatonin, which makes you sleepy, right? It controls your sleep-wake cycles and your circadian rhythm. So you might see that more in the sheep, because the sheep, what does sheep do really? They don't really talk, as far as I know. All they do is sleep, chew, smell, and make little sheep, right? Pretty much what I do my whole life. So pineal gland is endocrine. Endocrine means that the contents of that gland is going into the blood. Exocrine means it's going into ducts. Not ducts like Donald Duck, but in duct, D-U-C-T, in the body or out of the body. Okay, so again, now we're going back to the cerebrum. So let's just learn the parts of the cerebrum. I'm going to go through these with you, like frontal lobe, occipital lobe, parietal lobe, or three main lobes, but there's another two, right? I'm going to talk about the precentral gyrus and the postcentral gyrus. So you best know what I need you to know about these two, and I'll explain that as I show you the picture. So notice that the lobes of the cerebrum, this is just the cerebrum, which has a cortex, which we do our thinking and our voluntary motions, or muscle contraction, somatic muscle contraction is voluntary, and that comes from the cerebral cortex. And where we perceive somatic sensory and special sensory, we perceive them in the cerebral cortex. So that's where I'm going with pre and post central gyrus. But notice that they follow along with the bones, right? Occipital lobe is in the back where the occipital bone is, and the parietal lobe, which matches the parietal bone. So the parietal bone, occipital bone, and the frontal bone protect each one of these lobes. Don't forget there's two sides, there's two hemispheres. So there's a right frontal lobe and left, there's a left and right occipital lobe and left and right parietal. So we'll get back to post-central gyrus. And don't forget the temporal lobe. Where's the temporal lobe? That's another one. And the insula, but I'll go through them with you. So here's the temporal lobe. Insula is not on this page. I don't see it, but I'll explain where that is. Because you can't see the insula because it's deep to the temporal, parietal, and frontal lobe. You'd have to cut those lobes out. Septum pellucidum, I mentioned, is that membrane that separates the two lateral ventricles. All right, pineal body or pineal gland, we'll see. We're going to go through the diencephalon has three parts. The epithalamus, the hypothalamus, and the thalamus. And the mammillary bodies are on the ventral surface of the diencephalon, which you'll see. And then the interthalamic adhesion just connects the two parts of the thalamus. This is a good list. And then the brainstem, of course, the medulla oblongata, midbrain, and pons. Okay, and then we're going to talk about the superior and inferior colliculi, which together are this part of the midbrain. This is part of the midbrain, and that's called the corpora quadrigemina. Because it has 4 parts. It has 2 inferior colliculus and 2 superior colliculus. So together they're called colliculi. And this is also called the tectum. And that's part of the midbrain. Midbrain, which the midbrain is part of the brain stem. And then you'll see the cerebellum. Remember the tree of life, which is the arbor vitae, which is the white matter within the cerebellum. The cerebellum also has gray matter too. Okay, cingulate gyrus, I'll show you where that is. The fornix, these are all important. Olfactory bulbs, olfactory tracts. Of course, the central sulcus, very important. I'll show you where that is. And longitudinal fissure. Longitudinal fissure, again, in words, I have to show you a picture. Longitudinal fissure is just the groove between the left and right cerebral hemispheres in the middle. So it's above the corpus callosum, and I'll show you that. Transverse fissure, and of course, corpus callosum, the infundibulum. You're going to see what they cross over. The optic nerve, this is cranial nerve 2 crossover. Chiasm means crossover. You'll see that. The optic nerves, which again is cranial nerve 2, and then the optic tracts. So we'll go through all those parts. So this is a pretty good picture here of the top of the brain, right? And mostly cerebrum. So this is where I'm going to do this, I think. Let's see what's on the next picture. I'll do both. So we're looking at the top of the brain. Here's anterior. So here we have your frontal lobe, right? Remember I showed you kind of had to tell what's the front of the skull, right? Kind of tapers, and the back is kind of wide to tell you that's what's posterior and what's anterior. So that kind of helps you. Okay. Now, here's where I'm going to go. Pay attention to this. This is really important. I'm going to do this right here, and I'll do it on the next page. So first of all, this fissure in the middle, right here, and I guess this is what number 5 is pointing to, is your longitudinal fissure. Now, if we were in school, like in class, in lab, we'd be able to pull these 2 hemispheres apart because there's nothing connecting it on top. You could stick your finger through the pia mater and basically pull the 2 hemispheres apart. But deep in here is the corpus callosum. You'd have to cut that. That's dense tissue. It's neurons, it's axons, but you couldn't pull that apart. You'd have to cut through that corpus callosum. So the corpus callosum is deep to the surface of the brain. Of course, it's in the middle of the brain. So another sulcus, and this is really important. So there's a sulcus that separates the frontal lobe from the parietal lobe. Just to show you where the parietal. And this sulcus, number 3 here, is called the central sulcus. Makes sense, right? So pay attention here. So the central sulcus separates the frontal lobe from the parietal lobe on both sides. So this gyrus, and it's drawn in red here, it's the same gyrus on both sides. It's left and right, right? Left and right, correct. Because this is left and this is right. This is left and this is right. They're just showing a colored and non-colored brain. Colored helps though because it shows you the separations. So this gyrus, number 2, is in front of the central sulcus, part of the frontal lobe, and that's called the pre-central gyrus. So behind the central sulcus, on the parietal lobe, is the post-central gyrus. And you should have heard of this in lecture. Okay. And number 7 is just simply the occipital lobe. You can't see the cerebellum on this picture. okay? So cerebellum would be underneath because we're looking at the superior view. So that's really important right here. So the precentral gyrus is your primary, not association, primary somatic, which means like skeletal muscle control, motor area. So when I think about moving my pen, I'm using my, and I'm right-handed, right? I'm right-handed. So I'm using my left precentral gyrus to start that action potential, because it's all about somatic motor, moving my fingers. I can decide to do that. So the cortex of the frontal lobe, specifically the precentral gyrus, will start the action potential for me to voluntarily contract my hand and arm to write. But it crosses over, and that's called decussation. It starts on the left, the outcome in the PNS is on the right, because those tracks of neurons are going to cross over, and that's called decussation, right? So at the same time, I'm feeling the pen with my fingertips. I can feel the pen, and that's sensory. So motor is outgoing from the brain out, and that is efferent. And the effector is my skeletal muscle, my hand. Efferent is motor out of the brain to the PNS, out of the CNS to the PNS. Postcentral gyrus is where I voluntarily, not voluntary, where I cognate what I'm feeling in on my pen or from my fingers. So the postcentral gyrus is the primary somatosensory area of the cerebral cortex, of the cerebrum. So I'm going to cognate what my pen feels like right here. Not exactly here. You'll see, you learned in lecture about the homunculus, how there's different parts of the gyrus that cognates different parts of the body. So, but I'm going to cognate the feeling of my pen in my parietal lobe, specifically the postcentral gyrus. So you see how they drew it in blue and red? So blue is generally sensory in your book. Afferent coming into the CNS. Afferent is sensory into the CNS, where efferent is red, drawn in red usually in your book. And that's efferent coming out of the CNS, from the brain out to the peripheral. And that's my story. Really important. So in the same time, you can still see simple stuff like this is a gyrus, and this groove is the sulcus. Right, this is the longitudinal fissure. This is the central sulcus right here. Right, so here's the central sulcus. Not so easy, right? Not so easy when you don't have the colors, my friends. All right, and I won't do that to you. I might do something like this, like where I'll point to the sulcus, and I'll say, "What is that groove?" And you're going to type it in, central sulcus. Just like that, and because you wrote it so perfectly, you won't even get it marked wrong because spelling. Right. So remember, this is the frontal lobe here. This is here's the occipital lobe. Here's the parietal lobe. So here is your postcentral gyrus. And here would be your precentral gyrus. And you go through the list in APR and go through the different lobes. They're highlighted, and they'll be highlighted on the exam. Like this lobe is the temporal lobe. Here's the cerebellum. Right, and the spinal cord starts here. So above that will be the medulla oblongata. And here's where our big hole is, the foramen magnum would be. Right? So again, we could start labeling this, but this is not exactly the way you're going to see it. Like again, this is the full frontal lobe here. And here's all parietal lobe here. And this is occipital lobe. So it'll be specific or colored and highlighted. So the hardest thing sometimes is just knowing the difference between the cerebellum, which is here, and the occipital lobe. And I'll give you a mid-sagittal when we look at stuff like the medulla oblongata and the diencephalon you can't see. Now, if I cut away this area, right, like right in here, deep to the three lobes, the frontal, parietal, and temporal, you have a lobe that's deep. It's like the fifth beetle, right? You have four main lobes and one extra lobe. Poor Ringo. So deep to these other lobes is what's called the insula. Okay, insula. So you have your frontal lobe is here. And this is the left hemisphere, right? And this is all cerebrum I'm talking about. When I say lobes, it's about the cerebrum. The insula is a lobe of the cerebrum. This, like, it looks like a thumb, right, in a right-hand boxing glove, even though this is the left hemisphere. But it looks like the boxing glove thumb is the temporal lobe. And back here you have, oh, this is parietal lobe. And here's your occipital lobe. So I just wrote in the all about the cerebrum. And you have to know this is the cerebellum, but I don't want to write it here because I want you to know this is all cerebrum I'm pointing out, right? So in lecture, you remember like that the frontal lobe is mostly about motor, right? So in the precentral gyrus is about motor, primary somatic motor area. There's an area in the brain called Broca's area, the frontal lobe, which is about motor speech. Frontal lobe, motor. Broca's area is motor speech. And also, you have an area in here called the prefrontal cortex. And this is where we make our executive decisions, right? Am I going to get the spicy chicken sandwich today, or is it going to be the regular chicken sandwich at Popeyes today? Your big major life decisions come from the prefrontal cortex, right? Prefrontal cortex. And this is the part of your brain that really doesn't develop until you're in your early to mid 20s. When all those synapses finally decide, okay, we're finished making synapses. We're going to stay where we are and hopefully be able to make decisions on all those years that we went through. So this, of course, the sheep, I'm not sure if they have this area. We humans have this prefrontal cortex where you make all those decisions. Now I'm saying cortex, which is cell bodies. I don't know how this happens. This is the ghost in the machine. I don't know how you start muscle contraction, how we cognate, how we make decisions. But our decisions are based on memories and whether it's good or bad. So sometimes the insula has some input because the insula can be part of the limbic system, which is an emotional part of the brain. Right? But the insula is more about taste and smell and the memory of that. So there's connections between all of these parts of the brain, and those are called association fibers. Remember, commissures are connecting two sides of the brain or spinal cord. Then you have the temporal lobe is more about associating smell or hearing, right? And taste too is cognated in the temporal lobe, right? The parietal lobe is more sensory too. Remember, this is where the postcentral gyrus is, which is postcentral gyrus is in the parietal lobe, which is the somatosensory area, primary. You can have association areas just off of those to kind of integrate all the information that's coming in because there's so much information. You know, what you see, what you smell, what you hear, the memory of all of this, you know, and the thought and decision making, all has to kind of be put together. Now, occipital lobe should be the easiest because occipital means the vision. So your occipital lobe is what correlates and cognates what you see, what colors you see, fine visual acuity, you know, sharpening what you see, and resolution and clarity, all associated and cognated in the occipital lobe. All right, the cerebellum, there's none of that cognition, none of that volition. It's all about associating and projecting help with muscle contraction, right, which you'll see, and which you learned in lecture. Okay, now the bottom, this is the inferior view, and we're going to go through this in APR, but you're going to see, like from the bottom, of course, you have the spinal cord, and here's the foramen magnum. This is the cerebellum. Okay, this is going to be your pons, which is part of the brainstem. And this is medulla oblongata. Right? So, and you can see this is the frontal lobe here. And technically, you can't really tell, but this is the temporal lobe on the bottom. We're looking at the inferior view. All right, and this right here is the occipital lobe. When I say lobe, I'm talking about the cerebrum, just to make sure you know that. Okay, now you're going to see this structure right here, which is right below the frontal lobe. And these two structures sit in the cribriform plate. Remember that, of the ethmoid bone, separated by the crista galli, right? So these are your olfactory bulbs. Really big on the sheep. They got big olfactory bulbs. It's like I said, all they do is make little sheep and smell and chew and sleep, basically. So, but we have the same thing. We have the olfactory bulbs. Of course, smell is not as important. I mean, we'd miss it if it wasn't there, but we'd have to know it. So this one now, I'm going to say this number 8 here is the, I'm going to say olfactory nerve, but it's also part of cranial nerve 1. And technically, this is the olfactory tract. But I'll tell you why it's called tract, but it's still part of cranial nerve 1. Because below your cribriform plate, like into your nasal cavity, remember the superior nasal concha and the superior nasal meatus, you have the nerves, the olfactory nerves coming through the foramina of the cribriform plate. And there are special receptors for chemicals that we perceive as smell. So cranial nerve 1 is the olfactory nerve, and it's special sensory because smell is a special sense. But look where it is. It's way up here in the frontal lobe. So this has nothing to do with the brainstem. So cranial nerve 1, and you'll see cranial nerve 2 come off of the frontal lobe, basically, and diencephalon. But the rest of the cranial nerves, you can't really see them too good. They're so small. But there's cranial nerves that are coming off the medulla oblongata and off the pons, which you'll see close up later on. Okay? So right here, you have 2 nerves. And if you remember the optic canal of the orbit, well, this is where this nerve goes through, and this is cranial nerve 2, which is also special sensory, and that's the optic nerve. Not coptic, optic nerve. Optic nerve. Right? And this crossover part, because it's going to cross over, it's going to decussate. That's what crossover means. The crossing over part, get this word. Here's another cool word. Get ready. Get ready. This is the optic chiasm. That means crossover too, but it's decussation, which is crossover. So behind the optic chiasm, you have the optic tract. So to understand the difference between nerve and tract, like the optic nerve comes from the retina, which is the back of your eyeball. You learn that in AP2, special senses. So the retina has photoreceptors, rods and cones, which send the signal of depolarization to the optic nerve, which comes out of the eyeball. Then the optic nerve is going to cross over in an area called the optic chiasm. You have to know this for our practical, what the optic chiasm is, what the optic nerve is. So behind the optic chiasm, now the projection fibers of the optic nerve will go back to the occipital lobe to tell us what we see and colors of what we see possibly. So that behind the optic chiasm is the optic tract. So all of this on top of this is diencephalon, which you'll see later. So the back of the diencephalon, the bottom, I should say, sorry. The bottom of the diencephalon are these little bodies. And these are about taste and smell too. Remember, taste is probably 2/3 smell, olfaction, right? So if you had COVID and you lost sense of smell, it probably wasn't from your taste buds, but more from your olfactory nerve, because that's respiratory area, right? Nasal. Anyway, these two bodies right here below the optic chiasm are called mammillary bodies. And it's right under the diencephalon too, right under the hypothalamus. So again, this has an input for smell and taste. Again, the sheep, this is all this guy is about, or girl, smell and some vision, but not like our vision. Our vision is very complicated, very specific, crazy, right? So this is temporal lobe in here, the thumb of the boxer glove. I'm saying lobe, so that's cerebrum. Know the 5 lobes of the cerebrum, including the insula. Righto, okay. All right, so I think that's it. All the important things, you don't have to name. This is probably the diencephalon underneath there. That's all they're pointing out. So again, let's go through some of the parts. So I already showed you the main parts and the lobes of the cerebrum. So let's rule out the cerebrum. This is all cerebrum here. All that cauliflower looking stuff up here. You know where the corpus callosum is. You know where the septum pellucidum is. You know where the fornix is. Here's the corpus callosum here. Now, just one more thing I have to show you. Because you have to know the difference between this corpus callosum, and here's the septum pellucidum, or the ventricle, whatever you say. But this gyrus above the corpus callosum is called the cingulate gyrus. Sorry, it's an I. Cingulate gyrus. Okay. And you can see all the cool stuff, right? You can see the arbor vitae of the cerebellum. This is the cerebellum. This number 23 here is your 4th. You can spell it out or write it like this. 4th ventricle. This right here is the midbrain. Right here. And the back of the midbrain, right here, is your corpora quadrigemina, which has the superior colliculus and the inferior colliculus on both sides, because this is the mid-sagittal section. Okay, so that's part of the brainstem, and the cerebral aqueduct is here. Here's the pons, and here is the, I gotta say it, medulla oblongata. And here's the foramen magnum. And below the foramen magnum is where the spinal cord starts. So here's your 4th ventricle, which is a space for CSF, cerebral aqueduct. Your 3rd ventricle is right here behind the diencephalon. So let's do the diencephalon. Right here, this outlined area is the biggest part of the diencephalon, and it's number 6, is the thalamus. The thalamus is a relay station for all afferent sensory information except smell. Go figure. So everything that's sensory comes into the thalamus and is projected to the cerebral cortex. Very important. Right here is the most important part of the brain, I think, that we don't think with, and that's the hypothalamus. Please know where that is. And let's see where this is pointing. Right here is the infundibulum. I can't see the pituitary gland on here. But remember, the infundibulum connects the hypothalamus to the pituitary, so technically the pituitary would be here. You see it on the sheep sometimes, but not in the dissection. You'll see it in pictures. Now behind the thalamus, or above the thalamus, is an area called the epithalamus. That means on top, epithalamus, hypo is below, so this is the epithalamus. And the pineal gland is off of the epithalamus, which they're not really showing. Oh, maybe this is the pineal body right here. And there's just one little, like you see this little, it's like a little bump, but it's a connection. So I call it the interthalamic adhesion. It might have different names, but it's between the thalamus. It's a connection of the two sides of the thalamus. That's all that is, a connection. It's not just connective tissue. It's nerve fibers. So know the three parts of the diencephalon and where they are. The thalamus, which is the largest part with the interthalamic adhesion. The hypothalamus, which connects to the pituitary via the infundibulum, right here. So thalamus here, hypothalamus here. Epithalamus is behind, and the epithalamus, the top of the thalamus, is connected to the pituitary gland. Remember, the pineal gland. So the pineal gland, epithalamus, melatonin. Pineal gland is endocrine, not nerve tissue. Pituitary gland connected to the hypothalamus, endocrine tissue, not nerve tissue. Okay. So again, your list, and we should see if there's anything else on that list. The brainstem, the three parts of the brainstem are the midbrain, which is here and has the cerebral aqueduct in between it, really. So there's back and front. The back is the corpora quadrigemina with the superior and inferior colliculus, midbrain, pons, and medulla oblongata is here. Okay, the cerebellum, which has gray and white matter. The white matter is the arbor vitae. Okay, let me just stop, take a break for a minute, give you a break. Let's see. Cranial nerves, now there's 12 pairs. So again, this is going to be a bit of memorization. So this is a lab, so you're going to have to know, you have to identify them. And also, I'm going to be asking you questions about certain things like, are they motor? Are they sensory? Are they both motor and sensory? Are they autonomic? And are they special sensory? So let me repeat that. First, you have to know the names of all 12. And then you have to be able to tell me if each one is motor, purely sensory, or is it both motor and sensory? So M for motor, S for sensory, B for both motor and sensory. And then you have to tell me, is it a special sensory nerve? And I'll tell you the special senses. And then the last thing is autonomic. Does it have autonomic function as well as somatic function? And you know what that means because you finished with this in lecture. So let me just give you some examples. Like the easiest one is always the first one and the second one. Sometimes they get confused with these two, so be careful. So cranial nerve 1, again written in Roman numerals, is olfactory nerve. And the olfactory is smell, so that's a special sense. So it's sensory, yes, but it's also special sensory. Is it autonomic? No. Right? So smell is a special sense, olfaction. Number 2, cranial nerve 2 is optic nerve, which is sensory, but not only is it sensory, it's special sensory, because another special sense is vision. Okay? And then we have taste, right, which is called gustation. Now, this is a little more complicated, as you knew from lecture, of what nerves, cranial nerves, are sensing taste in your taste buds. So most of the taste, like 2/3 of your tongue, is facial nerve. So facial nerve is special sensory for taste, which is gustation. But the facial nerve is both motor and sensory. So you got to be careful there. And it's autonomic. All right. So another nerve for taste is glossopharyngeal. That's the back of your tongue. The back third of your tongue is special sense for taste, gustation. So this is also gustation. And the book doesn't tell you, but the vagus nerve is also taste, but not on your tongue, near your epiglottis and your uvula in the back of your pharynx. So vagus nerve is special sense too. Vagus nerve is the craziest of all the cranial nerves. It has so much function. And it's A and S too. In fact, the glossopharyngeal is autonomic nervous system as well. Vestibular cochlear is both. So this is special sense as well, because cochlear is hearing and vestibular is balance. So those are all your special sense nerves. So cranial nerves. So the special senses are olfaction, taste, which is gustation, and vestibular balance, and cochlear is the same nerve, cranial nerve 8. Okay, and vision is optic nerve, special sense. So olfaction, vision, vestibular, cochlear, which is hearing, and taste, which is gustation, are all your special senses. And number 3 is ocular motor, and you'll see it comes off the top of the pons, kind of small. So ocular motor is purely motor, but it also has autonomic function, ANS. And again, there's more tables which tell you more. This is just telling you about the word. So you have to memorize the number and the name. Right? So that's motor, but it's ANS because autonomic, because this is where your pupils dilate and constrict. Ocular motor. Trochlear is very boring. It's just one muscle that it innervates. You'll see the superior oblique muscle of the eye for eye movement, the extraocular eye, but it's just motor. Trigeminal is both motor and sensory, right? This is important for the sheep because this is all about chewing and feeling, around your eyes and face and jaw, right? Trigeminal. And it has 3 branches. The eyes branches ophthalmic, cranial nerve 5-1, maxillary is your face, and mandibular is your jaw. So that's why they call it trigeminal. And it's a thick nerve, easy to see, comes off the pons, not bad. Right, so below cranial nerve 2, the cranial nerves come off of the brainstem, starting at the midbrain, pons, and medulla oblongata on the bottom. Abduction is very boring, just motor, right, for a lateral rectus muscle of or one of the rectus muscles of your eye movement. So it's just motor. Facial is crazy. It's both motor sensory, autonomic, and special sensory. So your muscles of facial expression are controlled by your facial nerve, and that's cranial nerve 7. Okay. Cranial nerve 8 is purely sensory, but special sensory because it's vestibular cochlear, so it has 2 branches. Once it gets into the ear area, it goes to the ear, of course, for your cochlea in your ear for hearing, which is in your inner ear, and vestibular, you have your semicircular canals, which you'll learn in AP2, is about vestibular sense, which is balance or equilibrium, special sense, sensory only. Glossopharyngeal is a little tricky. It's mostly sensory. It does have some motor to one salivary gland. So technically it's both. I'm going to have to say both here. I don't care what the book says, but it's both. So if that's on the exam, of course, I'll give you all the credit you need. So it's also special sensory for the posterior third of the tongue. And it's autonomic because it has function in receptors for blood pressure. Very important. Now, the most autonomic nerve is cranial nerve 10. And autonomic, of course, is sympathetic, parasympathetic. But I'll tell you right now, all of these cranial nerves are mostly parasympathetic, especially the vagus. The vagal control is always slowing heart rate down, parasympathetic, decreasing blood pressure, parasympathetic, decreasing respiratory rate, parasympathetic. And so it's both motor and sensory because it also controls motor function at the heart and the digestive system. They call it the vagus nerve because it's a vagrant. It goes from your brain all the way down to your pelvis on both sides too. All these nerves are paired. And cranial nerve 11 is just motor. And it's only motor to 2 muscles. One is the sternocleidomastoid, remember that one? And the trapezius. That's all that does. In fact, it used to be called a spinal accessory because it was more like a spinal nerve, which we'll talk about later. It takes a long time. Okay. The last final cranial nerve is cranial nerve 12, which is only motor. And tongue, or glossus, means tongue. All right, so the glossopharyngeal nerve, cranial nerve 9, also is motor to the tongue, and pharynx, which is for swallowing. Right, swallowing is called deglutition. Right, gustation is taste, swallowing is deglutition. Crazy. So you're gonna have to go through these, and you're gonna have to learn each cranial nerve, and if it's motor, sensory, or both. autonomic, and special sensory. There's only 4 autonomics. There's the oculomotor is autonomic, the facial is autonomic, the glossopharyngeal is autonomic, and the vagus nerve is autonomic. Okay, and you know the special senses. I went through them before. You know, gustation, olfaction, vision, vestibular, and cochlear. So put them into the right nerve. So here is a picture you could use. I think this is a good one just to show you where the nerves are. So once you know the nerves, and you can tell me motor sensory, both at A and S or special sensory, then you have to find them, of course. And here's the olfactory bulb, cranial nerve 1, which is not off the midbrain, it's frontal lobe. Then you see the optic nerve, the optic chiasm, optic tract. This is a blood vessel, the internal carotid artery. And now you could see, you can't really see the midbrain so much here, but you could see where the nerve, the top nerves are coming from. So cranial nerve 3 is off of the top, more off of the pons, right? Oculomotor nerve, it's kind of hard to see, but it's there. That's cranial nerve 3, and the cranial nerve 4 is really small. Probably never ask you that one. But trigeminal, I will ask you, because that's a big one, right? It's a thick one. This is not a great picture. MH Connect does a much better job. Abducens is here, see that? And they're paired, don't forget it's on both sides. So here's where things get sticky, because they're very close together, like where the facial nerve is, and then the vestibulocochlear, cranial nerve 8. And you could put the name or the number, it's okay. Glossopharyngeal, and of course I'm going to ask you the vagus nerve on the exam for sure. And the spinal accessory has like rootlets to it, which you'll see again. And I'm not using this particular picture on the exam, I'm just showing you how they sequentially go downward. And then the hypoglossal nerve, you can see on the bottom. So I'll go through MH Connect with those cranial nerves with you later. So now the sheep brain. Again, you can see the dura mater. Huge olfactory bulb in the sheep. See the covering, the dura mater, it's like, you can't even see through it, it's so thick. It's like a bag, really, when you cut it off. Big, thick trigeminal nerve, right? Pituitary gland, too, is pretty big, because that's all they do, make little sheep and chew with the trigeminal nerve. Feel the food in their face and chew it, that's it. Here's the medulla oblongata and the spinal cord. So those are some familiar parts. You can clearly see the optic chiasm, right? Optic nerve, and you know the optic tract would be back here. So some general views. This is pretty cool because it like shows you they took the shell of the dura mater off, right? The falx cerebri is like part of the dura that goes in between the longitudinal fissure. So when you do a dissection, you actually have to cut the dura mater. It's that thick. And then you could peel the PO off and separate the two hemispheres through the longitudinal fissure. So this is like a shell of dura, like a shrimp shell. It's almost that dense. Inferior view, again, you could see same things, basically. I'm not going to ask you about the diaphragmacella. So that's just a portion below the pituitary. But trigeminal nerve, you should know, off of the pons. Pituitary gland. Optic chiasm is probably the easiest thing. There, an olfactory bulb. Can't miss that. Remember, this is the frontal lobe. This is temporal. And here. This is like an inferior view, and this is basically the spinal cord. It's kind of cool because it's showing you the dura and the trigeminal nerve. It's not showing you anything else. Like the arachnoid matter is actually connected to this. So it's nothing really to ask there. Same thing, basically. You can see the corpus callosum is deep to the longitudinal fissure. So this is, you could peel these apart only so far. Then you have to cut straight down through this to see a mid-sagittal view. You can see the temporal lobes and everything we saw already. Now this is tough. This is a tough one. Now in lab, we used to do this. We used to actually dissect the cranial nerves so you could see them. You see the cut infundibulum there, so there's no pituitary. So it's been cut out. Here's the mammillary bodies, just ventral or below the hypothalamus. Oculomotor nerve, right? You barely see it there, but it's there. Trochlear nerve, look how small that is. Trigeminal nerve is thick. And these are where it gets tough. I have to do APR and highlight each one of these. And you're just gonna have to memorize those things. So this is the hardest part, the cranial nerves for this exam. By far. Nothing else is this hard. And the accessory nerve, I would never ask on a sheep. So here, this is really important. And again, I don't think I'm going to show you this picture exactly, but you need to know this. Like, this is the back of the sheep brain, like the top of the sheep brain. And looking on top, you can see the longitudinal fissure. There's no more dura mater, so you can see like the shiny pia mater. Here's the cerebellum. Here's the spinal cord. So if I separated these two, like pulled the cerebrum forward and the cerebellum backwards like this, now you can see the posterior midbrain. And this is posterior midbrain. I'm just going to write midbrain, right? Posterior. This is really important too. Midbrain posterior view. And this whole area that I circled is the corpora quadrigemina. The corpora quadrigemina contains two parts. There's 4 bumps, right? The top 2 bumps are called the superior colliculi. Colliculus is singular, right or left. The superior colliculi is about visual input or visual reflex, I'll say. You might have learned this in lecture. The superior colliculus, now there's 2 of them, is auditory reflex. So the corpora quadrigemina is the posterior midbrain, contains 4 colliculi. The superior colliculi and the inferior colliculi. The superior colliculi is about visual reflex. The inferior colliculi is about auditory reflex. So what this means is, like, if you were looking at your computer, listening to this class, and you heard a sound, like a bang to your right, you would immediately turn your head to the right and maybe even lift your arms up, to protect your head. It's a reflex. And that input comes from the inferior colliculus, which is the auditory. Now, if you saw a flash of light, like a police car or ambulance light in your peripheral vision as you're looking at your computer, you would turn your head to the right reflexively, of course, with the help of the spinal accessory nerve, which controls the SCM and the trapezius. So all that visual and auditory input, of course, has to be processed in the thalamus, which relays all sensory input except for smell to your cerebral cortex. So okay, that was just a garbage can falling, or okay, that flashing light was just a passing ambulance. So know where to find those parts is my point with lab. Okay, so you'd have to, of course, pull the cerebrum forward and the cerebellum backwards to see that, or cut it away, whatever you want to do in lab when you're in there dissecting the sheep brain. So now look, this is a mid-sagittal sheep brain. Now look, the spinal cord is horizontal, not vertical like in the biped, like us, because the sheep is a quadruped. So you can see everything else, the frontal lobe, rostral means top, or cranial, caudal means back, like towards the tail. But I'm not going to ask you that. So what jumps out at you here? Look at the cerebellum, right? The tree of life, the arbor vitae, right? You can see the bump. The pons is kind of like a bump right here, and that's part of the brainstem. So the pons means bridge, like literally bridge. And what it's bridging is basically motor information coming from the cerebellum to the cerebral cortex. Okay, so the midbrain is above the pons, right? Peduncles are just connections of the brainstem to the cerebrum. And here's the medulla oblongata, which is a slight bump. The pons is much more exaggerated. Here's the spinal cord. So their foramen magnum would be here. Right? And you can see here, it's a little thinner, but here's the corpus callosum right here. And here's the fornix down here. Okay, here's the thalamus. Mammillary bodies, which are below the hypothalamus, and the optic chiasm. Again, there's no pituitary gland, so this gets a little confusing, but on the optic chiasm is right below the hypothalamus. So here's your hypothalamus. Here's your thalamus. And the mammillary bodies are here. So this is a little tricky here. This would be your septum pellucidum between the fornix and the corpus callosum. Look how big the pineal gland is in the sheep. We don't have anything that big because that's all they do, right? They stand up, they sleep, chew, and make little sheep. Posterior midbrain, you can see this superior colliculus. Now, if you just said colliculus or corpora quadrigemina, you'd be fine. Because here's again, these are the lateral ventricles. The third ventricle would be here. This is the cerebral aqueduct, that passageway to the fourth ventricle, cerebrospinal fluid. And then the central canal continues with cerebrospinal fluid down the spinal cord. And remember, the sheep is a quadruped, right? So here's the pituitary gland, big pituitary gland right here. You can't see the infundibulum here, though. Okay, so this is even bigger. You almost never see this on the dissection. It's confusing because you don't know if you're looking at fat tissue adipose or the pituitary gland. This is, again, this is a sheep, so we're looking at coronal section. So here's the corpus callosum. This is the right hemisphere. This is the left. So I'm not sure where this is as far as, is it parietal or frontal? This is the pons here, not the cerebellum. Here's the third ventricle. Here's the lateral ventricles. And the top piece is the corpus callosum. So you're going to see that in APR. You're going to see a coronal section. Look at the white matter, the gray matter of the cerebral cortex. It looks brown, but it really looks gray when you get a human brain. And the cerebral cortex continues all the way around the cerebrum. Here's white matter. Here's the cerebral cortex, gray matter. And the fornix, too. You can see on here as well. The fornix is on the bottom of the ventricles. So this is the motor sensory cranial nerves, again, which you have to memorize the names. You have to see where they are on the models and the dissections. And you got to say if they're motor sensory or both, autonomic and special sensory. So these are good study guides, so you don't have to go back to your textbook too much if you're just studying for lab. This is just extra if you want to, go in and look at different conditions, but we really don't have time to go through that now. Again, we might have done this in lecture, but I'd be happy to answer any questions. Just it takes a really long time. And I'm not going to ask you questions about signs and symptoms of dysfunctions of the cranial nerves, but it's good to help you learn about what sensory and motor is for your lecture. Okay, so this is pretty good too because it shows you each cranial nerve, and you can see the cranial nerves coming through the cribriform plate right into the nasal meatus. So technically the cranial nerves are in the meatus, below the concha. So the bulb and the tract are in the brain. Not in the brain, but below the brain, because remember the cranial nerves are peripheral. Inferior view showing the optic nerve. This, we use a lot of neurons to get that information to the occipital lobe. And most of it crosses over, which again, you'll learn that in AP2. Here's the oculomotor. Again, has fibers going to the iris to control autonomic pupil dilation and constriction. Okay, so the oculomotor nerve also is motor for muscles. You don't have to know the muscles because you'll do that in AP2, like all the rectus muscles and the oblique muscles. You don't have to worry about that. Just know that cranial nerve 3 is the oculomotor nerve. It's motor, not sensory, and it's also autonomic for pupil dilation. Cranial nerve 4 is very boring, right? It just controls one muscle, right? The superior oblique. That's all it does. It's purely motor. And you have to find it on the inferior view. Trigeminal nerve's got a lot going on, so it's got motor and sensory, therefore it's both. It's got 3 branches for sensory, ophthalmic, so feeling, around the head and eyes, the maxillary around the face, mandibular around the jaw. And it's a big muscle in chewing. So this is both motor and sensory, and you can see it, which is really good. Cranial nerve 6 is, again, one of the boring nerves because it basically just innervates the lateral rectus muscle. Like, your eyes move in an H and also obliquely within that H. So it's very intricate eye movements that involve these cranial nerves. And again, AP2, we'll talk more about the physiology. So just know that the abducens nerve is just motor to one extraocular eye muscle. Okay, we're showing the facial nerve now. Now this is, again, muscles. This is both motor and sensory. Sensory and special sensory, because it's the anterior 2/3 of the tongue. Taste, gustation. It controls all of these muscles you learned, the orbicularis oris and the frontalis. So it's big in muscles of facial expression. Right? It's showing the parotid gland here as a salivary gland, but it doesn't control that. So I don't want to make you think that. It does control the submandibular, so it has autonomic release. It also controls the lacrimal gland, the tear gland that goes across. Remember, the lacrimal bone is the medial orbit. But the facial nerve controls autonomic functions with tear production, release, and salivary glands, but not the parotid gland. And it's sensory and it's motor, so it's both, and it is autonomic. So this is showing you the anterior tongue as taste buds. Again, we're not doing taste. We don't do that until AP2 lecture. And then the posterior tongue is glossopharyngeal, but also cranial nerve 10, also, which is the vagus nerve. That's one of my favorites. Parasympathetic, autonomic, but it's also, there's taste buds in the pharynx that's using the cranial nerve 10, the vagus nerve, for sensation, special sense. Gustation is taste. This is the vestibulocochlear, which has two branches, right? See, it's coming from there. It's mostly sensory. It's sensory, 100% sensory, except for one little tiny muscle, which I'm not going to talk about. But the vestibulocochlear has two branches. One is vestibular, which is vibration receptors, which is mechanical receptors for vibration, for balance. And then the cochlea is this snail-like thing that has vibration sensors for hearing. So this is the cochlear branch, and this is the vestibular branch of the vestibulocochlear nerve, which is cranial nerve 8, which is sensory and special sensory. Cranial nerve 9 is the glossopharyngeal. Remember, special sense to the tongue for gustation, posterior third. Right, it also controls some salivary production motor, parotid gland, the largest salivary gland. It's also sensory for blood pressure in the vessels, so it's autonomic as well. So the glossopharyngeal is both sensory and motor, because it controls some of the tongue and the pharynx and the parotid gland. And sensory, taste, gustation, and blood pressure. It's autonomic, special sense, and it's both sensory and motor. Vagus nerve is huge. Look how far down this goes. This is autonomic, parasympathetic. It's motor and sensory. So it's both. And it's special sensory because it has taste innervation as well. Cranial nerve 11, again, we talked about the SCM control and the trapezius control, voluntarily, of course, and startle reflex as well. Not just startle reflex, but other reflexes as well, involving the midbrain corpora quadrigemina and the thalamus. Cranial nerve 11, right? Motor only. Cranial nerve 12 is motor only. Motor only to the tongue and some of the muscles of deglutition. Okay, so again, this will help you with your cranial nerves if you want to go through this, but we're going to do this in APR. Okay, more practice that you can do within that PowerPoint. Let me just see, am I still sharing that? No. I'll stop recording for a minute. Spinal cord. Reflexes are on here, but for our AP1 practical or exam 3, we're not going to use reflexes. We're not going to do reflexes. So the important things here, and we could ask you questions about, the different levels. Now, there's the top of your vertebral column is called cervical, and so is the spinal cord. But the difference here is in the spinal cord, there's spinal nerves, right? Come out from the side. So in the cervical region, there's 8 spinal nerves on both sides. They're paired. So total, there's 31 pairs of spinal nerves, right? So there's 8 in the cervical, where there's 7 vertebrae though, right? Then you have 12 thoracic spinal nerves. Then you have 5 lumbar spinal nerves, down here. Then you have 5 sacral spinal nerves and 1 coccygeal. Okay, so there's 31 pairs, and they come out from both sides. They're very somatic, very nuts and bolts, very simple wiring when it comes to these things. And there's going to be plexuses, like there's a cervical plexus and then a brachial plexus. You say brachial because you have to control the arms all the way down to the fingers for motor, and there's all that sensation coming in. So these are all cross sections of the spinal cord at different levels. Notice how thick the cervical is compared to the thoracic, because the thoracic doesn't control any limbs. It's just the intercostal muscles, basically, and the sensory and the information that's mostly autonomic in the mediastinum, heart and lungs. Then it thickens up again down in the lumbar because they have the limbs that are controlled. And sacral are kind of small. All right, so you'll see different levels and different parts of the spinal cord. So we're going to have to go through APR to go through that as well. But notice like this H, it's like an Under Armour H. And there's kind of like a hole in the middle. The hole in the middle is the central canal, which is continuous with the 4th ventricle cerebrospinal fluid flow. So the H is the gray matter. And everything outside of that H is white matter. Now let's talk about the different parts of the spinal cord. The horns, when you hear horn, that's the gray matter. Like posterior horn, lateral horn, anterior horn is gray matter. In the back, white matter is called posterior funiculus, lateral funiculus, here's the word, spelling, and anterior funiculus, lateral funiculus. Same thing here. So when you're looking at the spinal cord, that cross section, you can see that. And this is all explaining the white matter versus the gray matter, and funiculus is white matter. White commissure is the crossover in the spinal cord, the white matter crossover. Then there's gray matter, which has anterior horn, lateral horn, and there's going to be a posterior horn. And the gray commissure is the decussation or the crossover of the gray matter. You'll see that again in APR with the gray commissure. This is all things you need to know. Anterior median fissure, central canal you already saw, and posterior median sulcus. Notice that anterior is a bigger separation, so they call it a fissure, and the posterior is a sulcus, so it's a little shallower. So if you see, like right here, this is the anterior median fissure, that separation, this thing in here, and then the posterior median sulcus. Yeah. And different parts of the spinal cord, whatever's on your list, you see. And these are very, just nuts and bolts, basically, just naming the parts of the spinal cord. But for the most part, we're going to do the cross section, where you can see the gray matter, the roots, and the ganglion, which I have to show you. So this is just about naming the levels and the paired parts. Let me see if we got a good picture here of the spinal cord. Not so great, this PowerPoint for spinal cord, but so I'm gonna have to use this. Okay, so again, here's anterior, and this is posterior. So you see the gray matter. You have the dorsal horns of the gray matter, the lateral horns, and then the anterior horns. This is posterior funiculus, anterior funiculus, and lateral funiculus. Cross section is if you're looking down. Here's the gray matter. Then this crossover is called the gray commissure. The hole in the middle of the spinal column, spinal cord, is the central canal. This is your posterior median sulcus, the separation. This is your anterior median fissure. Okay, so now you got to remember some things from your lecture. So again, I don't think we have a better picture here. Let me see. Maybe we got one in the end. Yeah, let's use this one. I like this. So this is a reflex, but I'm showing you basically how things come in and out. So I like this because the blue is sensory afferent, starting at like a sensory receptor in the skin right here, like a pain receptor. So the sensory neuron will bring in the information into the spinal nerve. Now the spinal nerve, you notice, is both sensory and motor. They use red for motor, right? They use red for motor most of the time when it's like an orangey color. So let's continue. Sensory in through the spinal nerve, peripheral. Then it goes to the back. This is the posterior. This is the anterior. So all sensory information comes in dorsally, posterior. Sensory, blue. Here's your cell body of a unipolar sensory neuron. And a group of cell bodies in the peripheral nervous system is called the ganglion. So this bulbous part right here is called the dorsal root ganglion. Okay, this is the spinal nerve. It could be any level. It's in the arm, so it's up in the brachial plexus. So the spinal nerve, a nerve is a group of neurons, bundle of neurons that can be motor and sensory, and they are. This is the dorsal root ganglion, which is a group of cell bodies, and it's only sensory cell bodies because sensory comes in the dorsal root. And here's the dorsal root, which enters the dorsal horn of the spinal cord, gray matter. And the interneuron is in the lateral horn, and that could, in a reflex, it'll synapse with a motor neuron, which exits the ventral horn to the ventral root. This is the ventral root. This is the ventral root. This is the dorsal root of the spinal nerve. So all the efferent information, motor, is coming out the ventral root of the spinal nerve from the ventral horn of the gray matter, cell bodies, unmyelinated fibers. And then go back into the spinal nerve to the peripheral, where it goes to the skeletal muscle. And you know that whole story. So again, here's the central canal, the hole. This is the gray commissure, lateral horn, posterior dorsal horn, ventral horn of the gray matter. Lateral funiculus, dorsal or posterior funiculus, anterior funiculus. This is the white commissure right in here, white commissure. That's the best I could break that down for you. And that's pretty much all you need to know for the spinal cord. And again, we're going to go to the APR and we're going to show you, because I think I have to be there for that just to help you along. And I may even record that. So this is Module 7 in the Anatomy and Physiology Revealed. And we'll look at the topic, and you could go through your checklist. And the back has a pretty extensive list. And let's start with the hardest thing, the cranial nerves. And we'll look at the inferior view. And we can go through them all, if they're all here. So again, olfactory bulb, optic nerve, fairly simple because they don't come off of the brainstem. Oculomotor, right up top, the pons there. And trochlear, kind of small, see it? And then the trigeminal is here. Thick one, that's kind of easy to see. And then the abducens kind of stands alone. So now we're getting closer to the medulla oblongata, and so most of the nerves will be the bottom of the pons or the medulla oblongata, and here's where they get a little close. So let's go through these slow. So the facial nerve is cranial nerve 7. So 6, 7. And then cranial nerve 8, oculomotor. Oh, sorry, that was ocular. Not cranial nerve 8 is the vestibular cochlear. So it's down here, sorry. So what was the last one? Was the cranial nerve 7, facial, vestibular, cochlear. Facial 7, vestibular cochlear 8. Then you have cranial nerve 9. So 8, 7, and then cranial nerve 9. So 7 is here. This is 9, which is smaller, you see, compared to the vestibular cochlear, which is thicker in the middle there. Cranial nerve 10, which I'll definitely ask you, is towards the bottom. So these two are kind of difficult, the difference between the vagus nerve and the facial nerve and the vestibular cochlear. Vagus, vestibular cochlear. Vagus, vestibular cochlear. And then cranial nerve 11, which is accessory. It looks kind of different because it has those, rootlets on them. Okay, and then cranial nerve 12, which is hypoglossal, kind of in the middle with the tiny little rootlets. So know the difference between the hypoglossal, cranial nerve 12, and the accessory, cranial nerve 11. So 11 and 12. 11 and 12. Okay? And those are pretty much the way I'll show them to you. Like, I don't think, we're going to go through this too much. Like, yeah, I think I'm going to stick to the inferior view for the cranial nerves. So that would be much simpler. And you have so much other things to do. So I think that's fine. Okay, and then the brain.
Professor.
Yeah.
I have a question.
Okay.
Go
ahead.
The app, the McGraw-Hill says that my thing expired.
Yeah,
But
let me stop recording here if we're going to talk about something else besides what we're supposed to be talking about here. So let's look at the brain. Let's see which is a good view. I guess all of them. Yeah, I wouldn't ask something like this, the dura mater, or like what a gyrus is, though. Insular lobe. There you go. So this is a superior view. And the lobes, occipital lobes, you have to know what's frontal and what's posterior. Here's the postcentral gyrus. All right, and the precentral gyrus. And where's the central sulcus? Central sulcus. So this is an important one right here. This is a good one. And we could also do it on the lateral view too. So you have to go through every view for the brain. I don't particularly like this one, but I would definitely take a look. This, again, we didn't really see this yet. Like, I wouldn't use third ventricle, but I would use the gyrus for this particular view, superior view, and the lobes. That's fair. Now let's take a look at the lateral view. And we have to go into different layers. So, of course, I want the mid-sagittal is best. And here, so now you can go through the list on the left and you can see like the hypothalamus, of course. Frontal lobe, that's really good. Yeah, I like the lateral view for the deep structures, like say the brainstem in general, and then the cerebellum. I'll probably spend a bit of time here with the questions. Cerebral aqueduct, right between the midbrain, and that's nice. corpus callosum. So like Rajuna and I were saying, or like somebody was asking about, when you click them, you see the check mark. So take a couple of minutes. It won't take long. Just check off the ones on your list. Starting on the back of your checklist, and there's really some good pictures. Neuron is going to be very easy, very straightforward most likely from the atlas. The the models will be the neuron, so that's pretty straightforward. But this is a little more complicated. Like dorsal root ganglion, I would never put that in this picture. But dura mater, you could do. Fornix, nice. Fourth ventricle, the lobes, the hypothalamus we saw. There's the lateral sulcus, which I'm not sure if that's on your list, but there's a good picture of it. The whole hemisphere, right? The whole cerebrum. You could just say cerebrum. Mammillary body right there, because you get the mammillary body, the pituitary, and the pineal gland, which kind of get confusing sometimes. And then the medulla, the midbrain, occipital lobe, optic chiasm, all good ones here. I would spend a few minutes here. Now the pineal, there you go, off of the thalamus, and then the pituitary off the hypothalamus in the sella turcica. Very good. Postcentral gyrus and precentral gyrus, very good. How would they show you the right cerebral hemisphere? Yeah, that's cool. Septum pellucidum. But if you said lateral ventricle there, you'd be correct. And thalamus, third ventricle. Of course, what I'm doing now is not comprehensive. You have to go through your book, but we're doing a lot. We're definitely doing a lot. We already did the cranial nerves, which is great. And what else? Let's see, the spinal cord. Let's see what we got for that. Overview. Now, and you also have animations to go through, but they're not too much. It's just the divisions of the brain, which is animation that's part of your assignment. So you have to do that. You'll be responsible for that. Then it does the ventricles and the meninges, so that's really cool. So the only thing you gotta have to ad lib a little bit is doing what I'm doing between this and the atlas. Very good. Let's see. Yeah, I'll probably use the model more for the spinal cord. Let's see if we can see anything else. Cervical region. They don't really give you that much view here. The dorsal root versus the spinal cord. But these I would keep this very basic. Ventral root. So let's go over to the models. And same thing. I probably won't take too many of these, but because I don't like the way this blues it out. But I want to see what the spinal cord has. Let me just pause this for a second. It might take a minute for me to find that. So this is the spinal cord models, and very good. Central canal. Yeah, this is good. This is usable. Gray commissure. White commissure. This is good. Funiculus, again, the whole path through, and it shows you the real anatomy, which is excellent. Spinal nerve, right? Make sure that's a spinal nerve, and then the dorsal root ganglion. Just the ganglion, the whole dorsal root, just the ganglion, the whole dorsal root. So that's pretty good. Let's see if there's anything else in here. Let me just pause and look for one more thing.